to continue with this, I mostly focused my research on eukaryotic plant pathogens using EDNA. In this case, I used 2 oomycetes and 2 fungal organisms, which are plant pathogens, and I'm going to talk to you about the approach I used for eukaryotic pathogens specifically. So just a little bit of introduction, this one slide introduction. And in this case, I'm talking just about diagnostics, past and present. And we know, and most of us know, that traditional diagnosis is molecular based. It might be either PCR or it might be also via protein interaction. I just wanted to mention a little bit of history. Like, first time PCR was proposed by Henry Elrich and Trochterman in 1989. Sanger sequencing, we use it here at Oklahoma State and probably in many labs as a diagnostic tool too. Specifically for fungi and oomycetes, we use the ITS 1 and 5 or ITS 1 and 4 as targets, and we amplify those and sequence them and blast them, basically. That's the approach we've been using for diagnostics mostly here. But there's this new alternative, massively parallel sequencing, we've been talking today, and it was initially proposed in medical research and also in environmental samples. So mostly for environmental samples, there are a few softwares that allows us to interpret that data, and in this case, we have to blast or pairwise align the whole output from sequencing against the whole NT or NR database from NCBI, which takes a lot of time. And after that, we can use programs like Megan, which parses the data, or we can use programs as Metaflan also, which parses the data, but this is like more specific for some organisms, like very widely known organisms. But Megan is widely used to try to identify or detect organisms in metagenome databases. The thing is that they have shown limited diagnosis scope for plant pathogens and specific strains. So in this case, we focus in specific pathogens because we have to design EPROFs that Tony and John have already mentioned. So the pathogens I focused on were 4, Phytophthora ramorum, Puccinia graminis, Phacophthora phacorici, and Pythium ultimum. So most of you might know, or some of you might not know, Phytophthora and Pythium are the oomycetes, and these 2, Puccinia and Phacophthora, are fungi. And these are just some genome sizes that they have now. Usually the EPROF design for these organisms takes a lot longer than bacterial and viruses because of their large genomes. So we obtain like hundreds of thousands of EPROFs, which are useful for the diagnosis. These 130 million bases is just an estimation they did. So this is not a real. It is a real, but it's just an estimation of the genome size. But all of them are like fully sequenced, and specifically for Phacophthora phacorici, since there wasn't a whole genome available, we used express sequence tags for EPROF development. So this one might show different results after all because of that development. So everyone talked about EPROF design, but I'm going to talk about like sequencing samples a little bit in this slide. So whenever we try to sequence the samples, Tony and John, since they have a small genome of their organisms, probably they don't have to go through the step of nebulization, which shreds the DNA or RNA before sequencing. So in this case, for eukaryotic samples, it's very important to shred or cut the DNA into small pieces. So initially we have a gel running like this, where eukaryotic genomes or my DNA would get as near from 12 kV to around 2 kV. And specifically for 454 pyrosequencing, the technology we used, I had to size the DNA with nebulization and get the DNA down to around 1 kV to 600 base pairs or 400 base pairs to allow us to sequence the sample. Well, and these are just the extraction methods I use. So it's a critical step to nebulize or to size the DNA before sequencing for eukaryotic plant pathogens or organisms in this case. Probably you already saw this similar chart, but in this case we did initially, prior the in vivo scenario of EDNA, we did an in silico scenario where we had mock sample sequencing databases, which are simulated sequencing databases. But after that, we got the best quality EPROBs and just select those and use them in an in vivo scenario, which is the real sequencing data. So here is just like usually we did many trials from 40 base pairs to 140 base pair EPROB length and test them with mock sample sequencing databases. And specifically for eukaryotic plant pathogens, we end up selecting EPROB sizes because of the effectiveness of EDNA with these sizes. We end up selecting 40, 60, and 80 nucleotides in length EPROBs. So after this, we have the pairwise alignment, which we would usually use with prior any analysis of data from any sample. So this would be the BLAST. And then this would be the output, which usually, if we do it against the nucleotide database, NT or NR from NCBI, it's going to be like probably 10 gigabytes. In this case, we don't have that big file. And then we use our analysis to call presence or absence, as Tony already mentioned. So we used a different approach in eukaryotic genomes because of transposable elements we might have in fungi. So we are not using reverse EPROBs. Instead, we are using another approach as a negative control. So this would be like the overall thing for eukaryotic. But the EPROBs in this case, I just wanted to compare probably like viruses and bacteria have like 1,000 EPROBs or 2,000 EPROBs, which limits a little bit their detection scope. The good thing about eukaryotic organisms is that we might get hundreds of thousands of EPROBs, which are specific for the pathogen, as we can see here. These are the 4 pathogens I work with, and whenever I was testing many EPROBs, I was able to get up to 200,000 EPROBs for all 4 pathogens. And in this case, today we'll be using 80 base pairs EPROB, because the longer the EPROB we use, the longer the time it's going to take the alignment and the EDNA analysis. So, and the shorter the EPROB is going to take less time, but it becomes less specific. So today we're going to be using 80 base pairs whenever we do the hands-on with eukaryotic specifically. And these are just some numbers I wanted to show you how a big genome can help in diagnosis whenever we are designing EPROBs. And after we have the BLAST output, and in the BLAST output, what happens is we have an EPROB, and in this case, let's say this is an 80 base pairs EPROB. So it happens that it aligns with many 454 reads. So these 454 reads we might have multiple reads from the same region, which allows us to have a EPROB depth, I called it. So eukaryotic genome allows more stringent alignment values, as I mentioned before, because we have many EPROBs and we have many targets for the EPROBs. So EPROB depth is determined by how many times the EPROB hits on a different target read with high quality scoring. And high quality scoring, we select the scoring from the BLAST output. And what I call a high quality match from this pairwise alignment, and this is the e-value we all are using, 10 to the negative 9, a percent identity of 100% and a query coverage of 100% I can use with eukaryotic genomes because I have many EPROBs. And accepted depths are from 2x to 4x, which means like 2x would be this EPROB hitting only on 2 reads, or this EPROB hitting on 4 reads. But this is not the maximum. 4x is not the maximum. I can go beyond that. There are some, and we will see in the hands-on that we might have EPROB depths of like 100x or 200x, which is very good. And now I would like to talk to you about like what's a positive and a negative match. So whenever we have this EPROB alignment, for instance, here we have the EPROB, we might have false positive high quality matches, which means like it's hitting on negative on reads that are not from the pathogen or reads that are similar to the pathogen. So these I call them false positive high quality matches. And these are called true positive high quality matches, which are the ones that are hitting on the true pathogen DNA. So this becomes, and all this alignment is done with the scoring I already mentioned in the previous slide. So we can compare these two and make a call for positive or negative. So I called it the twilight zone or T-zone because we don't know, specifically for eukaryotic genomes or eukaryotic organisms, since they share many genome portions with other eukaryotic organisms like plants, we might have some false positive high quality matches, even though we eliminated EPROBs that are redundant. So I call the twilight zone whenever we don't know if it's positive or negative. And I did an analysis with 2,000 mock sample sequencing databases to allow to, so those data, those output would allow me to know what's the mean number of false positive high quality matches I can get, which means that I allow some false positive high quality matches in a data set, like let's say for Phytophthora ramorum, I allow 25 false positive high quality matches, which means that if I have 25 high quality matches, I might be doubtful if this is positive or negative. If it's lower than 25, I might be sure that this is negative. And this is pathogen dependent, so it has to be an analysis for each pathogen. Like for Puccinia graminis, we can use one false positive high quality match as a twilight zone or T-zone. So whatever is higher than one high quality match, it's considered positive based on our analysis, or whatever is lower, which would be zero, it would be considered negative. And this is the organism with ambiguity, which means that it might have, it might hit on Phacophthora organisms, and Phacophthora might hit on ramorums, or Phytium ultimum might hit on ramorums. So this is like organisms with ambiguity that we have here that we use to compare. So this is the approach I used. As I mentioned before, I wasn't able to use like the reverse EPROB. I used also the shuffled EPROBs, which would allow me to have same GC content and avoid these transposable elements problems or the reversions in eukaryotic genomes. So whenever we end up with a small equation here, which is for calling a pathogen positive, for calling a sequencing output positive or negative. In this case, we have high quality matches, and these high quality matches are the ones that I'll get after all the analysis, which means that I might get 10 high quality matches. And then these are divided by the false positive high quality matches that are the ones that I already got, which are, in this case, those would be constant for each pathogen, the false positive high quality matches. And this would give me a call. So in this equation, if C is higher than 1, the sample sequence database is considered to be positive for the pathogen. And conversely, if C is equal to or lower than 1, the sample sequence databases would be negative for the pathogen. So this is a small chart how I showed you, where I'm showing the time it might take, specifically for eukaryotic plant pathogens, to analyze the data. And this would be the EDNA approach, in which in this case we use a blast step here, where we have to get an SFS file from 454 initially, or we can get a blast output. We have FASTQ files here, and then we have to trim those based on quality. And then we have FASTA files. Usually we can get FASTA files directly from the sequencer, so this wouldn't be a problem. We can get FASTA files. But in this case, usually the blast step would take usually 36,000 seconds as measured by one alignment I did with some eukaryotic genomes whenever we do against the whole nucleotide databases. Whenever we are using eProbes, specifically for eukaryotic genomes, it might take up to 3,200 seconds, which is like probably 10% of the time which it usually takes a regular blast. Then the data parsing, we use EDNA approach. In this case, I'm using this approach where I'm using the T-zone for calling positive or negative. But in this case, like software like Metaflam or Megan5 would try to parse all data and assign taxons to each of the organisms that we might find there. So total time we end up with 8,000 seconds around, and total time we end up with 36,000 seconds. You can convert 2 hours, and it will end up with probably 1 day there. So this is like massively parallel sequences. This is the critical part. Alignment part and the MPS analysis is the critical part where we can get stuck whenever we have like multiple samples and we have to do all these analyses. It's going to take longer if we use like the regular approach. After all that analysis with all my organisms, we had, these are real sequencing data, and these are showing for 40 and 60 specifically eProbe lengths, and these are high quality matches, and these are the false positive high quality matches which are constant. So whatever is higher than 1, in this case, let's say for Puccinia graminis, it's going to be positive. So the call is 144, which is higher than 1, it's called positive. And here, whenever we use Phytophthora phacariaceae, with 60 it's negative, and with 40 it's positive. So that's why with eukaryotic plant pathogens we can use multiple eProbe sets. So one might call you positive and one might call you negative depending on the alignment. Like for Phytophthora ramorum, we have 2,000, very positive, and all of these you see. With 60 also positive, with 50 multimum, positive and positive. This would be a table that we will get at the end of the analysis, and then it will tell us if it's either positive or negative for the specific pathogens. And as I was mentioning before, the traditional analysis for eukaryotic plant pathogens or other organisms is like the alignment against the nucleotide database or the NCBI database. So I'm just showing here some output from the same databases that I used and comparing it. Let's say, in this case, I'm using Puccinia graminis sequencing output. And this is a Megan picture output from this analysis. So as we can see here, it's assigning a number of reads for each taxon profile. As we can see here, this is supposed to be infected with Puccinia graminis. But even though it's infected with Puccinia graminis, we find no Puccinia graminis in the alignment here. So it is skewed by the number of sequences that we have available in the NC databases. As we can see here, Phytophthora ramorum or Phytophthora cinnamomi are the ones that are getting the higher number of reads. And this might be because of shared genes or shared sequences that they might have that they are similar, and it's assigning a wrong taxon number whenever we have Puccinia graminis. We might get some other eukaryotic like Vitis vinifera here with 800 reads assigned. So it's just a comparison, but whenever we're using EDNA, we use 23 high quality matches, which would be called positive in this case. Similarly, in Phytophthora ramorum, again, usually we have multiple sequences of Phytophthora ramorum in the nucleotide databases. That's because there are many studies about Phytophthora ramorum. So in this case, if we would use Megan5, we would have like 5K or 5,000 reads hitting on Phytophthora ramorum if we analyze via Megan. If we analyze via Phytophthora infestans, if we analyze via EDNA, we get Phytophthora ramorum, and it shows 50,000 eProbes or 50,000 matches, and we don't even find it here, Phytophthora ramorum. And for Phytophthora phacariaceae, we can see again, it is skewed by the number of organisms or by the organism that has the higher number of sequences online. So in this case, again, Phytophthora infestans and Phytophthora factoriaceae, while we had 127 high-quality matches for our study. So this is just a comparison how our system might help us to identify organisms that are not in the databases, and more specifically, instead of using more general approaches like Megan-5. In summary, after all this analysis, EDNA was able to effectively detect the 4 eukaryotic pathogens. And I didn't present here the ultimum Megan output because while I was running it against the nucleotide database of NCBI, it took like, I don't know what happened, but it took like 5 days, and it ended up creating a file of 30 gigabytes, which I don't have enough RAM in my computer to analyze. Because Megan is using your own system, you have to use your own system, so I don't have 30 gigabytes of RAM in my computer to analyze that. So the selected EPROs, another part of the summary, the selected EPROs were pathogen specific, as I was showing previously. The sequencing output quality control is fundamental to achieve a reliable diagnosis. Sizing of fungal genomes, I know mycete genome is critical before 454 sequencing due to their large genomes. I already mentioned that. And the EDNA efficiency is pathogen dependent, and preliminary bioinformatic assessment with mock sample sequencing databases is needed, which means that we have to analyze each pathogen statistically to see which EPROV link we are using, how many EPROVs are we using, what areas are we targeting, and then we get a twilight zone, and we can use those EPROVs specifically for EDNA. So with all that, I'd like to thank all the members that I've had during this project. Dr. Garzon is my main PI, Dr. Snyder, Dr. Megan, Dr. Hoyt, and graduate students. I have John here also. I'd like to thank him, and project team, Dr. Stovey, who's there, Jacqueline Fletcher, Dr. Melcher, Dr. Ochoa Corona, Dr. Berman, and Dr. Blackman, and NIMFAB and USDA. Thank you very much. That's all I have, and if you have any questions, please let me know.
Hi, we have a question here in Ottawa.
Yes, go ahead.
Do you know how infested needs to be the sample to be detected, because you have no amplification here? So does your sample need to be really infested to detect it in the sample?
Well, whenever we collected the whenever we infected the samples, we infected them in our lab, and it wasn't really like completely infected. Specifically, let's say for, I didn't work with Phytophthora infestans. I was just showing a comparison against Megan and EDNA. I used Phytophthora ramorum, and I got DNA from Maryland in this case. And the plants were not really infected, but we take were infected, but we take the sample from a leaf that was showing symptoms. But whenever we are using EDNA, we're getting, instead of getting Phytophthora ramorum, we're getting Phytophthora infestans. I haven't developed EPROs for Phytophthora infestans, but I think that might have some problems if we are comparing with Megan, because Megan will show anyway Phytophthora infestans as positive in any eukaryotic samples for fungi because of the skewed data that we have. So, yeah, go ahead. Francisco here.
I'm sorry, this is Francisco. I'm chipping in into the discussion and question. Your question has to do basically with the sensitivity. And this is one of the things that we are working on. And basically what is happening is this. In theory, it's one thing, practice is the other one, but in practice we're supposed to receive a sample, and in the case of, the sample is going to be treated, and somehow the DNA or RNA extracted and then enriched. So we are trying to cope at the moment the low levels of DNA available by the enrichment. But still, we need to do probably more or additional testing with more precise sensitivity or serial dilutions just to get into details of the sensitivity of the method for particular pathogens, say viruses or fungi. I'm not concerned about the case of fungi because usually you can retrieve a lot of DNA out of there, but viruses are a concern. I don't know if that answers your question.
So it's really about sensitivity. And do you think if you use another sequencing technology that gives you much more sequences, so you could also improve the sensitivity there?
So I'm going to chime in here. This is Bill Schneider. What we did do was a whole lot of simulated in silico simulations to try and determine what pathogen read abundance you needed to have to be able to detect various pathogens. And for the most part, correct me if I'm wrong on this, but it was between 0.5% and 5% was the lower limits of detection for all the pathogens we looked at. So if you have between 0.5 and 5%, if you have 5% of your reads as occurring from the pathogen, that was sufficient for EDNA detection in every type of organism we looked at.
Thanks.
I have another question here from Ottawa. This was Guillaume Bilodeau, by the way, and this is André Levesque now. I think you should know there's 15 people here, so it was a very interesting presentation so far. And my question is around the probe length. I'm trying to get my head around that and the results you were showing. So if I understand correctly, when you have 80 MERS, and let's say, and then you have 40 MERS, so you would have all the possible 40 MERS within the 80 MERS, and would that explain to a certain degree why you have so many more hits with the shorter ones? And is it also a combination of the fact that when you have shorter ones, is that you're picking up the reads that have got only the partial part of your probe? So the shorter the probe, the more likely, or you increase the likelihood of picking up partial reads. Is that? So I'm trying to get my head around these results. Could you maybe explain a bit how you have these results of big changes in reads, or in the hits? Sorry.
Big changes. Usually whenever eProbes are developed, usually the program which uses is like MOMER and NUGMER, which is a SNP. It will find for SNPs, and it will develop eProbe from different lengths from that SNP. So it will select that SNP, and then it will start cutting different lengths of eProbes, which would give me like 20, 40, 60, 80. But in this case, whenever we are talking about e-values or pairwise alignment scoring, in that case, the eProbe length matters, because whenever I was using 20 base pairs, I wasn't able to use e-values because e-value won't allow me any whatever is 20 base pairs will hit on anywhere. So which means that I was detecting false positives. So all that analysis was done with many mock sample sequencing databases, that, as I mentioned before, to tell me that I have like the proper eProbe length, although those might be from the same area or same genome region. I have to select the proper eProbe size, which is pathogen dependent or organism dependent.
I think